Okay, welcome to my first video. Today we're going to be talking about mitosis. The purpose of mitosis is pretty much for growth and repair. So if you get a cut or you're just a little baby and you need to get bigger, mitosis is how one cell turns into 2, turns into 4, turns into 8. And mitosis is split up into different phases. So the first phase we're going to talk about is prophase. And at this point, the long strands of DNA called chromatin, they condense into things called chromosomes. Now, chromosomes have a short head here. I don't care if you can see this video, is for me, not you. But they also have a centromere in the middle, and they have a long head on the bottom. And those heads are called telomeres, and the one in the middle is called the centromere. Anyway, so in order to separate in the cell, they need to get rid of this nuclear envelope because they can't separate if there's this wall. So this nuclear envelope is going to dissolve in this prophase. So this is going to go away. And that brings us to our next phase, which is metaphase. Up here we just have some keywords that I learned. And in metaphase, the chromosomes line up in the middle. They are getting ready to separate. So we have these things on the outside. I don't really know what they look like. These are called centrioles. And connected to the centrioles are these fibers called fiber spindles. And they connect to the centromeres. They're getting ready to pull apart. So that's what happens in metaphase. In anaphase, that's when the chromosomes actually get pulled apart. The sister chromatids, we like to say, which is pretty much just these two parts right here. This right here is one sister chromatid, this is another sister chromatid, and they're gonna get separated into their own chromosomes. So in anaphase, they start to get pulled apart. The sisters get separated. I don't really want to draw it out. Pretty much, there's the cell, and the sister chromatids get separated. Okay, and in telophase, they are completely on the far sides of the cell. I'm drawing the centromeres there. And these things are so close to separating, we just need one more phase. And that is, oh, this is telophase, sorry. They're super close to separating. At this point, the chromosomes that are super tightly packed, they start to loosen up a little bit. They turn back into chromatin because they're going to be just like they were before when they were just one cell. And so that's pretty much it. In cytokinesis, the cytoplasm, the wall of the cell, splits off into 2. And we have all the same fun stuff that we did before the cells separated. Okay, at the end. Hope you learned something. Actually, I don't care. I learned something. That's all that matters.